presenting this on behalf of a group from UH Hilo. PI is Marta Di Maintenon from Marine Science. I'm co-PI from the same department. But I want to give loads and loads of credit to our master's student, Holly Jessup, who has really spearheaded this project, and this is really her baby. She's giving a presentation at SIGB conference in Texas this week, so she cannot be here to present this. There's been a great deal of research that's been done on sea urchin herbivory in Hawaii with a specific focus to Trypnustes gratilla done by Stimpson et al. At UH Manoa. And then their work has shown that the grazing by Trypnustes can make a significant impact on macroalgae in Hawaii, especially, well, including invasive algae, such as Gracilaria over here. So what we wanted to do was to investigate the feeding ecology of another genus of sea urchin, Echinopteryx, and their impact on keeping the invasive algae down in Hawaii. So just to give you a little bit of a background on these Echinopteryx species, sea urchins, okay, there are two nominal species of Echinopteryx, diadema and calamaris. Okay, both are very, very abundant. We found them up to like 3 sea urchins per square meter in Hawaii, but these guys have been relatively little studied, especially around the Big Island of Hawaii. They belong to family Diadema today, and they have wide distribution ranging across the Indo-Pacific to nominal species. There's more story to that. I'll mention it in a minute. And they can get monstrous. I don't know if you've seen them in the water, but they can get, if it's 15 centimeter test size, then with the long spines that they have, they can be kind of basketball size monsters. Okay, so what we wanted to find out was their feeding ability, like I said, in comparison to Trypnustes, and our project had 4 components. Okay, before we launched into the feeding ecology and feeding behavior of these guys, we needed to find out the diversity within this genus, Echinopteryx. Okay, and then we did a couple of experiments to investigate their feeding behavior. By tank experiments and using stable isotope signatures, we determined their relative trophic positions relative to Trypnustes. And then we also did a whole bunch of underwater surveys to determine their abundance and distribution in relation to each other and in relation to where the algae were found. Okay, and we collaborated with Dr. Bob Nishimoto at DNR for that component especially. Okay, so I'll tie them all in together at the end, and I'll also show you some of the outreach activities that we've been doing with this project. Okay, so to give you a little bit more background on Echinopteryx, there are 2 nominal species, right? Like I said, Diadema and calamaris. But there is a little complication to this because, as you see here, you can find 2 different color morphs of Diadema, the white morph and the black morph. And even the 2 species, Diadema and calamaris, when it comes to the black morph, are very, very difficult to distinguish, especially underwater. Okay, they look very, very similar. Okay, so our question here was how many species of Echinopteryx exist in Hawaii, and what does each species look like? Can we make a key to identify them separately? Okay. Hypothesis here is that sets of physical characteristics distinguishing Echinopteryx morphotypes, meaning the Diadema and 2 color morphs of calamaris from Hawaii, are indicative of separate genetic identities, separate genetic phylogeny. Okay, so to do this, we collected 75 Echinopteryx specimens from 12 sites all around the Big Island of Hawaii, and additional 13 specimens collected at Kingman Reef by NOAA folks, which is about 1,400 kilometers southwest. That map is a little bit tilted, southwest of the Big Island. And we brought them back to the lab to do the morphometric analysis, and we used primary set of 3 morphological characters to first designate them into the 2 species, Diadema and Calamaris. Okay, and I'm just going to show you them. But the first criterion we used was that in Calamaris, in both species, you see the primary spines that are thicker, and then the secondary spines that are finer, needle-like spines, right? The secondary spines in the Calamaris are not banded. The primary in this specimen are banded, but the secondary spines are not banded. And in Diadema, however, in this species are striped, banded, as you can see here. Okay, the second morphological character we used was that the primary spines this time of Calamaris, you see that the transverse or horizontal ridges, right? Whereas Diadema, you see the vertical ridges going along. Okay, and then the third characteristic was that that we used was that the apical portion of the test, the aboral apical portion of the test, is raised into a cone in the Calamaris species, whereas, and sometimes in the earlier, in the younger specimens, you see what's called anal sac attached to the apical portion, and Diadema, the apical portion is always flat. Okay, so once we categorized them, ID'd them into those species, we also used a secondary set of morphometric characteristics to see if we can differentiate between the black and the white morphs of Calamaris. Okay, so these are the secondary characteristics we used. Symmetry, or the shape of the genital plates. These are the little plates at the top of the apical portion of the test. So what we used was the ratio of the height and width, basically designating the shape of the genital plates, and also genital plate length, the size of genital plates in relation to the overall test diameter. And to kind of give an indication of the shape of the test, we measured the vertical height of the test in comparison to the diameter. Is it flat or tall and round? Okay. And then the last characteristic we used was the size of the peristome, basically the mouth opening of the test, in relation to the overall test diameter. And none of these characteristics turned out to be significantly different between the two color morphs. And this is just showing the data from the first characteristic here. Okay, so this was a morphological evidence. Well, this morphological data showed us no difference between the two color morphs of the Calamaris. Okay, so we looked into genetics also. We looked at 539 base pairs section of mitochondrial DNA in all of 88 specimens collected, sequenced both ways, no cloning. And this is what the genetics showed us. Okay. You don't really have to worry about any of these up here. But the main story here is that there is deep and clear divergence between Echinothrix diadema and Echinothrix calamaris. And so basically, all the diadema specimens were monophyletic, all the calamaris specimens were monophyletic with each other. Okay, so there was a clear genetic and morphological distinction between the two species, but when it came to the difference between the color morphs, genetics again showed us that there was no difference. In fact, 4 of the 8 white morph specimens we had actually shared the same haplotype, unique sequences with other black morph calamaris. Okay. So for this section of the project, we're confident, well, we can, first of all, we devised a set of morphological characteristics that could distinguish between the two species of Echinothrix found in Hawaii, and they can be, yeah, and then there is no significant difference morphologically or genetically between the two color morphs. So they're not good candidates for cryptic species here, which has been suggested actually from French Polynesia specimens.